It gives me a great pleasure to introduce Dr. Lori Swikert today. She got her undergraduate degree in psychology from the University of Tampa, her PhD in biology from the Florida Institute of Technology, and then did postdoctoral positions at Duke University, Florida International University, and Max Planck Institute of Chemical Ecology. And she recently joined the faculty at UNC Wilmington, which is my alma mater, and how we connected because I saw that she was putting out an ad for graduate students, and maybe you'll let us know if you're still looking for graduate students. And the ad caught my eye because it was my alma mater and her research sounded really, really cool. So I invited her to come talk to us today. So welcome, Dr. Swikert.
Thank you, Terry. Can you hear me?
Yes.
Great. Can you see my screen as it should be?
Yes, looks great.
Thank you both, and thank you all for coming. Certainly an honor to be able to present today. And yes, as it was mentioned, I just accepted an assistant professorship at the University of North Carolina, Wilmington. I am recruiting both for this coming fall and the fall after that. So if you have any interest in light, life, and color, that you will want to maybe apply to my lab, okay? So in short, my work combines neurobiology and sensory ecology to ask larger questions about marine animal vision. Okay, and I study marine animal vision in the marine world because the unique pressures of the light environment here drive innovation and diversity in marine animal vision. So we're familiar with light above water. There's lots of light up here, lots of colors for our eyes to use, and light on a day-to-day basis is fairly predictable. But there are different rules for vision underwater. And that's because underwater light is highly dynamic. Okay, and that's for 3 reasons. And if you're a scuba diver or if you've ever snorkeled, you may know some of these already. The first reason underwater light is so dynamic is because when sunlight strikes water, wavelengths are separated out by energy. So low energy, long wavelength light is absorbed first, and high energy, short wavelength light penetrates to depth. Second reason is ecological factors. So you have turbidity and algal blooms that can shift the intensity and color of underwater light. And then thirdly, you have this great variation in habitat complexity, from the detailed appearance of coral reefs to the uniform waters of the midwater pelagic, right? And so together, these factors have put pressure on animals, marine animals, pressure for them to tune the sensitivity of their visual systems to these requirements, and to adopt an array of visual phenotypes, okay? And when we study this visual diversity, we can then ask questions about the principles by which these systems operate, their evolutionary underpinnings, and their resilience to environmental change. And so my research program has been varied, different questions and different systems, taking me from new frontiers in the brain and in neural tissues to new frontiers in deep sea biology. I'm going to talk about a couple of these systems today that really sum up my program. The first, I'm going to start with the North Atlantic right whale. Okay, so what started as a question of right whale conservation quickly became a fundamental question about how mammalian retinas can be designed. So lessons from a retina like no other. Then we're going to leave the eye. I'm going to talk about phototransduction in skin, all right, skin vision, and how that relates to the ability of the hogfish to rapidly and dynamically change skin color. And lastly, I'm going to talk about my most recent work, looking at variation in bioluminescence in deep sea shrimps. Okay, so a lot of storytelling. So before I can tell you any of these stories, I have to tell you some background on vertebrate vision and realize that what I'm about to tell you here also applies to your own eyes, okay? So vision begins when light enters the eye and is focused by the lens on a neural tissue called the retina. The retina is a beautifully organized tissue made up of several cell layers. The cells that absorb light are photoreceptors. There's two types of these, rods and cones. That's because rods are shaped like rods and cones are short and conical. Here, cone. Rods support dim light vision. They function at very low light levels, such as at night or at great depth. Whereas cones function in bright light conditions, daytime vision, and cones can give you the ability to see color. So logically, what that means is when you're only using your rods, you can't see color. So you can try this tonight in your bedroom, you go to bed, after about 20 minutes, your eyes will adjust, and that adjustment period is your rods coming online, okay? And you look around, your brain knows what color your walls are, your brain knows what color your bedspread is, but if you're actually looking, you can't see color. So it's interesting. After the photoreceptors absorb light, they pass information to retinal interneurons called bipolar cells. And that information is received by rod bipolar cells and cone bipolar cells. And then they give information to ganglion cells whose axons make up the optic nerve. Looking closely at photoreceptors, these cells have two parts, a nucleus containing inner segment and a light-sensitive outer segment. And that outer segment is sensitive to light because of a special molecule called visual pigment. Visual pigments are made up of a 7-transmembrane G protein called opsin, okay, and a vitamin A chromophore. And it's this relationship, okay, of opsin to chromophore that determines the wavelength or color sensitivity of your photoreceptor cells. All right, so the last fundamental piece of information to know is that there are different families of opsin proteins, one of which is associated with rod photoreceptors, the rhodopsins, Rh1, and there's 4 families associated with cones. And they have to do with different wavelength sensitivity, like short wavelength sensitivity or long wavelength sensitivity. And your expression of these opsin proteins determine your spectral sensitivity or light sensitivity. Okay, don't worry, I'm gonna walk you through it. So in 2011, as a graduate student, I attended the biennial meeting for the Society of Marine Mammals. And I met Dr. Jeffrey Fazek. And from that meeting, we started this collaboration where we thought if we can study cetacean vision, maybe we can reduce their entanglement. We had this idea that if we could understand what these whales could see, perhaps commercial fishing gear could be designed so that it would be more visible to the whales in the water column, that they would be able to avoid them earlier. And we decided to focus on North Atlantic right whale vision, both because this is a critically endangered animal, and also because at this point, their visual ecology was unknown. And so the first step in this project was to understand what opsins are expressed in the right whale retina. And we understood all cetaceans, okay, dolphins and whales, to express one type of rod opsin and one type of cone opsin, okay, in their retinas. And so we went on the hunt for these. And in the North Atlantic right whale, what you're seeing here is the sequence of the right whale cone opsin aligned to a control, the bottlenose dolphin. And bottom line is, when Jeff sequenced this, you're seeing the amino acid sequence, he found an early stop codon, a truncation mutation that essentially cut the cone opsin in half. Oops, so this renders cone photoreceptors not functional, leaving only rod photoreceptors in the retina. This is a condition known as rod monochromacy. At the same time, so this was surprising, okay, we're going to talk about this. At the same time, there was a group at UC Riverside doing a genomic survey of cetacean opsins. What you're seeing, and they expanded on our results. They validated and expanded our results. So what you're seeing is a phylogeny from that work, Very quickly, the blue lines indicate blue light sensitive cone opsin. So you can see that was lost very early in cetacean evolution. The red lines indicate red sensitive cone opsin. Okay, just a longer wavelength cone opsin sensitivity. And what I want you to see, 5 times independently across cetacea, cone opsins have been lost. So we have rod monochromacy in the ballinid whales, which is right whale and bowhead, the rorquals, sperm whales, and beaked whales. This was shocking for 2 reasons, truly. The first was, up until this point, all mammals were thought to have both rod and cone photoreceptors, something called a duplex retina. Both strongly nocturnal animals and fossorial animals. Secondly, we understood that similar deactivation mutations leading to the loss of a major photoreceptor type causes degradation of the retina. It was thought to be only in pathology that we saw this in mammals. So for example, people who were born with retinitis pigmentosa have a mutation of their rod opsin gene. And basically after birth, after some time, because the rod photoreceptors are not functional, you have degradation of the photoreceptor outer segments, both rods and cones, apoptotic loss of all of those photoreceptors, and rewiring of the inner retina. So how is whale vision sustained? Is it even sustained, right? So this led us to the question, how does the evolutionary loss of cone photoreception affect the circuitry of the mammalian retina? We did this with a histological study of vision using bowhead whale retinal tissue that we were able to procure from an Inuit hunt, and it's a closely related, a relative of the North Atlantic right whale, okay? So the very first question was, how was cone photoreception really lost? We wanted to make sure that cone opsin protein wasn't still in the retina. So you're going to see micrographs of cross-section of the photoreceptor layer. And we looked for the opsins using immunohistochemistry. Very simply, we made proteins of interest glow. Using commercial antibodies, we made the rods glow red and the cones glow green. And first, you have to apply your antibodies in a positive control. And so we did that with a closely related land mammal, the domestic cow. And what you're seeing here, the blue dots are photoreceptor nuclei, and the red and green are showing rods and cones as they should be in a retina. When we applied this in the whale, we only ever saw rod labeling. We tried different antibodies, different regions of the retina, different concentrations, and we never saw cone opsin labeling. And this made sense. This was what the mutation would predict, that it was no longer being translated. Okay, so now the question was, if cone opsin is lost, are the cells still there? Are the cone cells still available? So this time we used light microscopy, and you're looking at a cross-section of the retina again. And this is in our positive control, the domestic cow. And you're seeing the nuclei of all the photoreceptors and 2 morphological types of cell as you expect. Those long cylindrical rods and the short conical cones. When we did this method in the whale, we saw only a single morphological type of photoreceptor, that classically of rods. Again, this is consistent with at least what we've seen in the pathology literature, right? It seems that with the loss of cone opsin, we've now had a loss of cone outer segments. But what about the mRNA transcript I told you about? I told you that Jeff Fazek sequenced cone opsin in the right whale, and we did that from mRNA. That suggests that a cone cell still exists transcribing that message. And so perhaps even if the outer segment has been lost, the inner segment has still been maintained. So we went on the hunt for that. Now you're going to see micrographs of the nuclear layer where we're going to look for those cone inner segments. No cow, we're just looking in whale this time. Based on the size, staining, and position of the nuclei and signaling structures, we can tell rod and cone intersegments apart. Bottom line, we found both the rod and cone intersegments. So this is how you can tell them apart. In mammalian retinas, the rod nuclei make up the majority of this nuclear layer. They're smaller, they're heavily stained. Whereas cone nuclei are always at this very top boundary, they're larger and more lightly stained. But beyond that, if you look down in the synaptic layer, it's a little hard to see, but you see this classical morphology of a rod sphereal and of a cone pedicle, which is this triangular bit. So it appeared that the cone intersegments, the signaling components of cones, were maintained. Okay, so then the last piece of the puzzle here is, have the cone bipolar cells been maintained? Remember those interneurons that receive cone message? So we shifted down in the cross section here to the bipolar cell layer. Again, using immunohistochemistry. And you can tell rod and cone bipolar cells apart by their immunoreactivity, so which antibodies work, and then morphology, position and morphology of the cell. Bottom line is that we found both a rod and cone bipolar cell field using antibodies against calcium binding proteins, this one specific to rod bipolar cells, this one specific to cone bipolar cells. We labeled both cell fields and then confirmed that they were each cell type by their specialized morphology. So this very long axon cell terminating by what are called ganglion cells here for the rod, and the different subtypes of cone bipolar cells, on and off types. So it appeared that the cone bipolar cell network had been maintained. The million-dollar question was, why maintain cone intersegments and cone bipolar cells in a retina without functional cone photoreception? Well, we believe it was to maintain all possible signaling pathways for rods. If these whales are only going to have rod-based vision, then they need to make sure that they have the greatest utility of that sense, that they've still maintained every way that we know rods can signal through the retina. Walking you through that real quick, the first way we know rods can signal through the retina is the classical pathway, when they synapse directly onto rod bipolar cells. And we know that this functions in extremely dim light conditions, so very light sensitive. But then over time in this field, we found that there's two other pathways. Rod-cone coupling, where rods synapse by gap junction directly onto cone pedicles, and then through here, and that functions at brighter light intensities. It lets rods still be functional as light levels increase. And we know that rods can synapse, this is the most recent understanding, rods can synapse directly onto cone bipolar cells. And the cone bipolar cell network is critical for visual processes such as center surround receptive fields and direction selectivity. These are underlying things in how vision works. And so this makes sense that you would maintain cone bipolars and you would maintain the cone intersegment here so that you can maintain this coupling pathway. So this hypothesis that remains to be tested is non-photosensitive cones maintained to preserve this functional array of rod-based vision, which was cool and informative, especially for again, in studies of pathology of mammalian retinas. But the larger question being whale conservation, if they don't have cones and they're just working with a single type of photoreceptor in the retina, what can we tell these rope manufacturers? Well, Dr. Fazek went on to look at the spectral sensitivity of rods, the color vision of rods. And this is what he found. So what you're seeing on the y-axis is how sensitivity, okay? And on the x-axis, you're seeing the visible light spectrum. The rods, the whole retina of North Atlantic right whale is sensitive at a peak of 493 nanometers. So what is blue-green light for their rod? And because they have no cones, in part, they can only see in black and white. These animals don't have color vision. What this means is that any light that falls under this curve, these colors appear white to them, and anything outside of it appears black. Okay? And so, ecologically, this kind of makes sense. Their prey, their favorite prey, this copepod, Calanus finmarchicus, has a red oil drop in its body. It's very red. And so likely this rod has evolved to match the background blue-green light of the coastal waters in which they live. And then they are visualizing clouds of these copepods as highly contrasted prey items against the background. So seeing what it appears to be black prey on a white background, okay? So now you are a conservation biologist. You have to tell the rope manufacturers what to do here. And what we would say is that based on their visual ecology, we need to be designing ropes that are highly contrasted according to their vision. So not white ropes that just reflect all the ambient light, not blue or green, but red or black ropes that will highly contrast in their eyes to background. Okay. So far, we've been talking about ocular vision, but there's a whole other side to vision and photoreception that I find particularly fascinating. That is extraocular photoreception, light detection outside of the eye. This is getting more heavily studied as time passes. We are finding extraocular photoreceptors across a great variety of animals and tissues. But we have less of an understanding of the function. Right now, we know it plays several roles. Extraocular photoreception across animals can support phototactic behavior, so responses to light, anti-predation responses, like shadow reflexes, and the ability to rapidly and dynamically change the color of skin. Okay. Now, I first got interested in this with my interactions with the Caribbean hogfish, a fish that we find off the southeast of the US and in other parts of the Caribbean. It's an animal that can change its color and pattern in seconds or less. So what you're going to see here is this hogfish swimming along the bottom, and it's relaxed in a very light or white coloration against some of this bright sandy background. And when a diver approaches, you're going to see it change color. So right there. They're called the hogfish because they root around on the bottom to forage, and that's what that animal was doing. So you can see those chromatophores in the skin, those color cells responding, this bright, like, get away from me, anti-predation coloration, and then swims off again. And so I first got this idea that the skin might be desensing light when I was recreationally harvesting them in the Florida Keys, okay? So I was capturing hogfish, putting them on the deck of my boat, and when I flipped the fish over, in this instance, flipped the fish over to put it in my cooler, I noticed that the side that had been facing the deck had turned white, like the deck, and had taken on the texture of the deck's grip. So to be clear, the fish was facing the other way, and I had just flipped it over before I took this photo. And so, in what was my naive perspective, I thought that it was seeing the deck and changing. It really amazed me. I mean, this told me that color change could happen post-mortem, and it could happen maybe autonomously, right there in the skin. Now, with a little experience, I believe that this pattern was probably because of pressure or temperature of the weight of the fish on the deck, but it gave me this idea. And I looked in the literature, and I found so much evidence for a relationship or data to support relationship between animal color change and skin light reception. Across color changing fish, cephalopods, amphibians, reptiles, we find that animals that can rapidly change color also have phototransduction of the skin. Now, a commonality of these animals is that they change color via specialized skin cells called chromatophores. Within vertebrates, this works by a rapid dispersion or aggregation of pigment granules within these cells. And so across these animals, if you take a preparation of skin and you put light on the skin, it has intrinsic photosensitivity. Color change will occur directly in response to light. If you then break up that tissue and send it out for sequencing, okay, you'll find that these tissues express the suite of phototransduction genes required for phototransduction in the retina. So they have this ability to sense light very similar to the retina in the skin. And I don't mean optically like vision. I'm saying it's just a photodetector, detection of light. And so we find this across animals, but the function is unclear. Based on some behavior, we believe that dynamic camouflage is thought to be mediated by vision. So the value of dermal photoreception to color change remains unknown. We don't know why these animals can sense light with their skin. And this is the major question of my work and my research moving forward. So we first attacked this by, in hogfish, because this hadn't been studied before in this animal, comparing phototransduction in the retina and skin. I did this with de novo transcriptomics, and I don't want you to get bogged down in the details, but know that this analysis yielded the largest transcriptome for hogfish to date, yielding about 28,000 genes in the transcriptome, which we annotated. Using this pile of data, we compared gene expression in retina and skin. So a little throwback to cell and molecular biology, the way that phototransduction works, just briefly, is that photon strikes an opsin. The opsin activates a G protein complex, which acts upon an enzyme that manipulates secondary messengers for cell signaling. Okay, so based on the data I had gotten from transcriptomics, all these components that you would expect to be in a vertebrate retina were there. 1 rod and 4 cone opsin types. So they had the opsins, the G protein complex, transducin, and that downstream enzyme you would expect. In skin, however, the only gene, the phototransduction gene, shared by the visual pathway in the retina was the SWS opsin in skin, or blue or UV sensitive opsin or detector in the skin. Then we found an alternate phototransduction pathway based on different G protein complexes and a different enzyme that works on cyclic AMP, which actually has been predicted in fish skin, color-changing fish skin, from other studies with using pharmacological experiments. And so this made sense to us, showing that there were separate but complete phototransduction cascades in retina and skin in the hogfish. And this was interesting because in a comparative sense, this was different from what we understand to be true in cephalopods like the cuttlefish. Cuttlefish have the same phototransduction components in retina and skin, where hogfish, and again, this is predicted in other fish, we see separate phototransduction components in retina and skin. It's just interesting to indicate that there was separate evolutionary histories to these phototransduction mechanisms. But the big question really, right, was what is the functional significance of the dermal photoreception in skin color change? And that's where we took it from here. So first, I just needed to show that in hogfish that dermal photoreception was actually connected to color change. And we did that by making those skin preparations and exposing it to light. So if you want to know what colors of light a retina can sense, you put an electrode on the eye and you flash different colors into the eye and measure retinal response. We did the same thing in skin. We flashed different colors of light on preparations of hogfish skin. And instead of measuring an electrical response, we measured the amount of color change that occurred. And this is what we found. These are the pictures of the different skin preps and the response to light exposure for 3 minutes at different wavelengths. So you have response on the Y, 2 wavelengths on the X. And not only did we see a response to light, okay, great, the dermal photoreception is connected to color change, at least in a artificial system, in the dissected system. We see that the skin differentially responds to wavelengths of light, with the peak sensitivity being over the blue end of the spectrum, which from the sequence of the SWS1 in the hogfish skin was the predicted peak just based on the sequence. So this made sense to us because we only found an SWS1 opsin in skin, which is blue sensitive, and it appears to have high blue sensitivity, showing that SWS1 is likely an important mediator in this response that we saw. Great. Okay, so this SWS1 appears connected to skin color change. So then the big question is, where is it in the skin? Is it all over on the top? Is it down deep? Is it in certain cells? Again, we used immunohistochemistry. What you're seeing here is a confocal micrograph of hogfish skin in cross-section. Here's a pigmented chromatophore, a melanophore, and the direction of light is coming from right to left. We applied a commercial antibody for SWS1 and found that SWS1 opsin was expressed just beneath the pigment of chromatophores. We looked for it again, different preps, and we found SWS1, it's very highly localized in its compact areas, expressed beneath this layer of pigment within the skin. So you see here, this white arrow is pointing at all the chromatophore pigment. So it's localized beneath chromatophores. So as a neurobiologist, I was like, well, this is interesting. It's a transmembrane protein. It requires a ton of membrane to be expressed at high levels, like this amount of expression. So to show you this, even in photoreceptors in the retina, they require so much membrane to house opsin that you see this adaptation of all these membrane stacks, right? And so I predicted that, well, there has to be quite a bit of membrane here if that's actually SWS1 expression. Well, we did TEM, and check it out. This is a chromatophore. This is the melanophore zoomed way in, right? So these are melanosomes filled with melanin granules. And right beneath it is a separate cell that is filled with reticulated membrane. We don't know what this cell is. It's a putative photoreceptor. We don't know if it's neural. We don't know anything about this. It's just, it is so fascinating because if this is a specialized cell for photoreception, then this would be the first time that a specialized cell for photoreception has been found outside of the vertebrate retina. And so a major goal of the lab is to investigate the properties of this photoreceptor. So yes, so this structure being potentially a scaffold for the opsin co-localized to the SWS1 expression in hogfish. So you might be asking yourself, why is the opsin beneath this chromatophore pigment? You think about, if it's there to look at the outside world and help an animal change color, why isn't it superficial on the skin? Why is it buried beneath a shield of pigment? So we ask the question, what is the pigment doing to the light? Why hide the photoreceptor beneath a shield of pigment? And we did this with spectroscopy. So really quick, you're seeing a scale of a hogfish that is white. When it's in its white coloration, all that pigment's aggregated. And when the hogfish is dark, all that pigment is dispersed. We passed light, white light, through individual cells. 60 cells per cell color, and of which there's 3 major types: the black melanophores, red atherophores, and yellow xanthophores. And you don't need a fancy graph to tell you that these are very red or long wavelength cells. On the y-axis, you see transmittance, on percent transmittance. On the x, you're seeing the rainbow, okay, wavelengths. And so yes, they look very red, and they are very red. They transmit long wavelength light, they let a lot pass, and they heavily absorb blue light. That's why things appear red, right? They're absorbing blue. And so, especially the red and the black cells. So the exact light that the skin photoreceptor needs for activation is the exact light that this pigment is blocking. Why put the photoreceptor behind a blocking shield? This is what we think. We think that dermal photoreception is providing sensory feedback about skin color change. We think it's a clever mechanism where the body can see its own skin, know how it appears, okay? Let me tell you how this would work. If when the hogfish is in its white coloration, the pigment is aggregated, and this would allow the light to pass and activate the opsin. But when the shield disperses, when the pigment's dispersed, we see that the pigment absorbs blue, and this would lower the activation of the SWS opsin. So maybe instead of this thought that I think that's been propagated in the field, that we think that the skin's looking at the environment and changing accordingly, at least in this system, it looks like it's embedded beneath the color change screen and it's watching its own color change. So this may monitor and help to regulate appearance. And this is important. I don't know how much time you spend thinking about sensory feedback, but it is interwoven into almost all physiological and behavioral aspects of organisms. You need checks and balances to make sure that you're performing correctly. That helps survival. So quickly, how do I know I have two fingers behind my back? Well, I have proprioception. I can feel it. I can't see it, but I can feel it. I have feedback that I've performed that correctly. In the same way, which is what is a life or death situation of color change and camouflage, it seems that these animals are monitoring their own appearance and perhaps regulating it to make sure they're performing correctly. So this is the next step of this study, to test this. And I'm going to end here with just a couple more slides just to talk about a recent incredible opportunity I got to go to see. In 2019, the end of 2019, kind of right before this pandemic, I got to go on the NOAA Ocean Exploration Research Expedition. It was titled Journey into Midnight. This was in the Gulf of Mexico. And I got to work with incredible colleagues, and we found this diversity of results, all centered on exploring visual ecology in the deep sea. Okay, so just looking at this graphic, I don't need to tell you that vision and visual cues appear important to deep sea biology. As light levels are lost, and light becomes completely attenuated where there's no traces of sunlight, you can see the different strategies for camouflage change. Trying to match to background, mirroring background, becoming transparent, and becoming heavily pigmented, then becoming less pigmented. And at the end, if you have any questions about why these strategies are employed, I'm happy to chat about it because I think it's fascinating. But what I want you to get out of this is that as sunlight is lost, bioluminescence becomes very important. Bioluminescence being the natural production of light, the emission of light by living organisms. And so our mission was to study the diversity and functional significance of bioluminescence in the deepest parts of the Gulf of Mexico. And so I got to experience this diversity firsthand with this gorgeous family of shrimps from the Sergestidae, genus Sergestes. And these animals, they heavily rely on bioluminescence using specialized light organs called organs of Pesta. These light organs are modified pieces of hepatopancreas. So these are pictures of hepatopancreae from different species, and these lobes are bioluminescent. What makes this group interesting is that the light organs have diversified to the species level. I'm saying each species has a distinct light organ pattern. It's so important that taxonomists use this character to identify this group. Now, in the 80s, these light organs have been implicated in counter-illumination. That's the emission of light from the ventral surface of an animal to replace the light blocked by the body when viewed from below. So when a predator looks up at these animals, they just blend to the downwelling light. But this predicted function of counter-illumination has no obvious requirement for the morphological diversity observed in this group, And so this led us to this question: have the light organs of the Sergestidae diversified for conspecific recognition? Are they using these different patterns to ID each other to the species level? And I would say this because these animals are both copulatory. We know that they come together in copulation to reproduce, and these species are sympatric. So it would suggest that using some sensory biology, they do need to identify each other before, so that they can reproduce. And so we did this fun little study looking at visual ability to basically see if these animals had the capacity to tell each other apart by bioluminescence, by their pattern. And so we selected 3 with very diverse patterns here: what is a bilobed, trilobed, and fringed morphology. And so we had 3 aims to the study, and the highlight of each of these is what I'm going to give you. But we first just looked for hints, looking for sexual dimorphism in eye size. This can be pretty common in visual biology, where one sex of a species will have a larger eye, indicating that perhaps that sex has to visualize the other for sexual signaling. So we looked for any clues there. Then we modeled bioluminescent sighting distance, the visual range that they might be able to see each other in situ. We tried to estimate, model the distance in which they could see each other's bioluminescence. And lastly, we gave these guys an eye chart exam, okay? We looked at the morphology of their eyes to estimate visual acuity, how sharp their vision is, and then if they could have the capacity to discriminate these fine patterns. So the take-home conclusion of each is that we did not see any sexual dimorphism in eye size investment. Okay, so using allometry across the sexes and species and ANCOVA, we did not see any differential investment in eye size. So no help there. It doesn't mean that they don't use it for sexual signaling. It just means that we're not seeing any evidence of that in eye size investment. Okay. Then we modeled bioluminescent sighting distance using a model that has been developed in 2014 and then modified more recently. And you put in different parameters of vision, bioluminescence intensity, and so forth. Happy to chat about the details later if interested. But for these guys, we estimated variables that we couldn't measure, including how intense their bioluminescence emissions might be in situ. It's hard to measure that in deep sea fauna because they die when you bring them up from the deep. But this is from precedent in the literature of known intensities of deep sea animals. And so there's quite a range here from 1/10 of a meter to, let's say, 5 meters that they might be able to see each other's bioluminescence against the black background. We could talk more about this, but despite that this is variable, it seems ecologically relevant. The mean estimated distance across here, which is just one metric here, is about 1.3 meters that they might be able to see each other, okay? We know from trawling experiments that these species can be as dense as up to 150 individuals per cubic meter. So they're able to see, they're interacting with each other over very short distances. And we know that based on related species like euphausids, they probably have really bright bioluminescence. So it makes sense. They can see each other within distances that they occur in nature. So that was promising. And lastly, it was time for that eye chart exam where we measured different optical properties of the eye. We got a metric of how sharp their vision was. We put it into R using this program, Acuity View. And what Acuity View does is it takes an image and it renders it as it may appear to an animal given their visual resolution. It transforms an image as it should be perceived given the spatial resolution of an animal. So we generated images of each species by tracing them, their bioluminescent organs, and by doing the morphometrics, we got their visual acuity estimate. And bottom line, this is what we found. Not great for the shrimp, right? So in the columns, you're seeing what this species looks like over and over again. Okay, it's just the same. This is all P. armatus. This is all A. sargassi. This is all D.N. Seni. And then the row is the visual ability of the species and how each image would appear to them. So they're comparing across row, which of course then the yellow asterisks show looking at a conspecific. And modeled at this distance is just 2 centimeters away is what's modeled here. We modeled this across different distances, and even closer, it doesn't appear like these animals can resolve the fine differences in light organ pattern. Right? Unless there's some kind of neural edge enhancement or some visual neural process going on to really sharpen these images. So this is what we think is happening, right? Organ pattern appears to have diversified for reasons other than conspecific recognition. So they may identify each, we know that they're likely identifying each other because they do find each other in nature. Maybe they're doing it through a temporal signal like flashing as opposed to the pattern, or using other sensory systems like chemosensation from these big, beautiful, long antennae. But the pattern diversity doesn't seem to be a visual signal. So this imparts caution about assigning function to traits we see based on human perception. We need to know perception of animals and to see if something has meaning. People have been hypothesizing forever that across the Sergestes that these were a visual signal, but the data doesn't support that, right? And lastly, so they may have diversified due to factors such as evolutionary drift. And this is just one hypothesis that the Bracken Grissom lab, my old lab, is now testing. All right. With that, I'd just like to thank my colleagues across these projects, my funding agencies. And with that, I'd like to take your questions.
Okay, thank you. I can see only one question in there. I don't know if the host can see more than me, but there's a question. It's like, if the right whale's food is red, I understand that they would be able to see it, but would they be attracted to anything that is red?
That's great. So the question being, would they mistakenly be attracted to a rope because it's so contrasted to background, like they would be attracted to prey? That's so wise, because we can't institute a solution for a problem that creates more problems in nature, right? Two things I would say. One is that the clouds of copepods are providing them other sensory information and appear very different as a dynamic moving blob, as would a static, sharp, vertical, very artificial structure in nature. And in fact, Dr. Scott Krause of the New England Aquarium has started to test these hypotheses by coloring different PVC pipes different colors and putting them out there in arrays kind of in front of foraging right whales with all the approval you would ever need to do such a study. And they do visualize these vertical stimuli and get out of the way. So they're, at least in what behavioral trials with these animals, they seem to be avoiding them as opposed to feeling attracted to these kind of things. But that was a good question.
Okay, there's a couple more questions in there. Are red and black roach being encouraged for use while by fishermen?
It's a great question. So are we encouraging the use of this? Yes. And in fact, the fishermen are on board. I'm talking about crab traps and lobster traps, especially through Cape Cod, where that's big business. And the fishermen don't want the whales to get entangled in their lines. One, because they care about the whales, and two, because if their tag is found on a dead whale, they're fined. That may not seem fair, but that's how it's. They really don't want their whales striking their gear. And so it's a beautiful thing, because this didn't have to be legislated and go through it. They're looking for this data, and just by hearing this, they're now purchasing ropes of these darker colors.
And I had a question, because I was just reading a paper today that was looking at bird collisions with structures. And they were making an argument because of where the eyes are located in the head, that the range where they have binocular vision, where they can have better depth perception, is so small that part of the reason they might be running into structures is this lack of depth perception because of the, you know, the binocular vision, visual range is so truncated compared to ours, at least. If you thought about anything like that.
It's a fascinating point. Yeah, right? Because you need the overlap of vision for stereopsis to see depth, right? For depth perception. Field of view studies in whales are really limited, but this is something we care about. Do their eyes face forward enough to have any part of binocular vision, or do they truly have kind of separate visual fields going on? That's not something that has been heavily studied, and we're looking into it. We're right now compiling a ton of cetacean vision studies so we can create a mass database of kind of what we know and don't know, and visual field has been really neglected.
Cool. Okay, gosh, it looks like there's a bunch of questions in here. So why might we not see this ocular evolution seen in whale and other marine animals?
Oh, okay. So why not this novelty in the whale and other marine animals? So what we're talking about here is this loss of cones, right? There's so many answers to this question. What I would say to you, and this is fascinating, and if you have interest in evolutionary biology, is the loss of cone opsin here, as published by this group, was due to a drift lost by chance over time, meaning they weren't selected against. That wasn't like bad for the animals and then it was selected against. It just was no longer being selected for. So that means that they were doing just fine with rods. And there's a lot of selection pressure on photoreceptors. Why? Because it's neural tissue, and neural tissue is expensive. It takes a lot of ATP and mitochondria to make them fire. All this to say, then why haven't other marine animals lost their cones? Well, likely because there's still selection to keep those cones going, whether that be for color vision or so that they could see in bright light. Since this study, the only other naturally occurring rod monochromats that have been discovered are the xenarthins, which includes armadillos and strange little organisms like that. And so that's a great question. Hard to know why we're seeing this novelty in the cetacea.
Thanks. What are some more potential applications of your research that you were excited about?
Oh, potential applications of my work in terms of, well, I'm going to just a quick little story I'll say about this, and it's to a colleague's work that I just found so fascinating. On this research cruise, I don't know if you guys saw the press like in late 2019 about a giant squid sighting in the Gulf of Mexico. That was this cruise. So I got to be out there where Edie Witter was living her dream, employing basic visual ecology science to capture the video of the first giant squid. So you talk about applications of this kind of work. The quick story is that she had been studying bioluminescence her whole life. She found the North American jellyfish does a burglar alarm bioluminescent signal with light is put out in a beautiful display over and over whenever the jellyfish gets attacked. So stay with me. So jellyfish gets attacked by a fish and it starts to bioluminesce like crazy. And the reason it's doing that is to call in bigger predators to eat the fish that's hurting it, right? That's the burglar alarm. So she created this Medusa technology, this yellow thing behind the team. That right there is the bioluminescent lure. She had a string of LEDs making the same color and pattern that the jellyfish does when it gets attacked. And when she deployed this into the Gulf of Mexico, we deploy this as a group. It called in a giant squid to be videoed for the second time ever in history, first time in American waters. So long story short, an application of so you're just studying why do jellyfish glow? And from that, you build technology that then helps us conserve one of the most elusive organisms on the planet, right? So that's just one application.
Okay. Is the SWS1 opsin specific only to hogfish, or is that same opsin present in other organisms like reptiles and cephalopods?
These are such great questions. These students, I hope so. I hope these are students asking these questions. Yes. So this gecko I showed has SWS1 in its skin, and it's fat, and that's the opsin that's in the gecko skin, and they're also really long wavelength. So this might be a conserved mechanism or some convergently involved mechanism for dermal photoreception. And so interestingly, flatfishes like the flounder have 5 opsins, and the cuttle that's even a whole nother mess. But the flatfish 5 opsins, so they might have the capacity for color vision of the skin, that they're doing some kind of wavelength comparison of the skin. So the next downstream purpose of my laboratory is to compare what I'm calling single channel dermal photoreception, so just like hogfish or this gecko, versus flatfishes that have multiple opsins and maybe more complex skin vision. And cephalopods are just unbelievable because both their retinas and their skin are thought to have black and white photoreception, right? They don't have different opsins, and yet they can match color extraordinarily to different backgrounds, and that's a mystery that persists in the field.
Cool. Okay, near the start of the presentation, you mentioned that lab assistant opportunities may be available for this research. Where could I find more information on something like that?
That's a great question. Please email me. Here. My email is swikertl@uncw.edu, and I have a lab website as well with my contact information.
Okay, so someone says, my Wi-Fi has been going out, so you may have answered this already, but it seemed to me that many animals in the deep ocean where there's very little light are colored red. Has this been studied to affect bioluminescent communication? And does this coloring help some animals avoid predation or become better predators or something to that effect?
I'd love to ask you what you thought, right? I'd love to ask you what you thought. So, absolutely. Let's talk about what's happening here. Basically, 2 things. First of all, there's no more downwelling light. And I told you red was the first to go in the water column. It gets absorbed really quick. So since there's no red light making it down here to reflect off these animals, it's an incredible camouflage. It's as if they're pitch black against background because there's absolutely no red light at this depth to reflect off of them. So then you ask, why aren't they just black? Well, when you look at physiological studies, it's metabolically less expensive to make red than black. So that's a whole nother field. Like, so you see red, you see black, you see a lot more red because it's cheaper. But the other thing is the problem down here. Why aren't animals just transparent then? Why aren't they just transparent at depth? Well, bioluminescent searchlights. There's a ton of fish that have flashlights of bioluminescence under their eyes shining out. They're looking for prey, or the anglerfish, right, with that bright lure. And because most bioluminescence is blue, those searchlights are ineffective against red because the blue is coming out and it strikes the shrimp, and then it just gets absorbed, not reflected because there's no red light in that bioluminescence. So bottom line, it is a master coloration for camouflage at depth.
Okay, would the color change pattern, the wording's a little weird, slow the escape process when being attacked?
Okay, I understand what they're referencing. So in that video, I showed this animal's changing color. The hogfish was changing color when it was afraid of being attacked. And maybe the question is, does that help them kind of wave off the attack or slow down the attack? We would think yes, right? So I don't know if you've heard of this concept, aposematism, but when you're warning a predator that maybe you're unprofitable to attack, you're dangerous, you're poisonous, you're venomous, or whatever. So this isn't necessarily true aposematism, but bottom line, what's happening with the hogfish here, they're typically matched to background with their color. But in a dire situation where they feel like they're going to get attacked, they throw up their dorsal spines and they put on this strike pattern to basically tell a predator, I'm big, I'm bad, you're going to get hurt if you try to mess with me. So the answer to your question is yes. I believe that this is an anti-predation response. Yeah.
Concerning evolutionary biology, you discussed the continued presence of cone inner segments in bipolar cells to maintain signal pathways. Is it possible, theoretically speaking, for whales to readapt to cone utilization if it were required in a different environment? So basically get it back, I suppose.
I guess so. I mean, I don't know how many things in nature have gone away and been re-evolved. Yes, I think that's entirely possible over evolutionary time. And what's fascinating, though, is then these animals would no longer share a common ancestor that would give them this capability. They would have to re-evolve the conopsin to not have that mutation.
Yeah.
So I think that's possible.
Yeah, I guess mammals did it because we all derived from nocturnal animals that lost a cone, and then the primates had a mutation that developed a new cone from that. So I guess it's possible.
Definitely. And notice here that the toothed whales, like the dolphins and some of the other toothed whales, maintain that cone. It's mainly the balinid, the baleen whales, and some toothed whales that have lost the cone. So there seems to be an ecological basis.
Yeah. So they might not get back the same thing. They could get something different.
Mm-hmm.
Yeah. So I had a question because we had a recent speaker that's a friend of mine that does sensory ecology, and he mostly deals with sound, and mostly a lot of how does sound pollution affect animal behavior. And so what he was finding in birds, which are mainly visual predators, that in loud environments, their ability to successfully attack and get prey was diminished, even though their visual system wasn't affected. And his thought was that it's just the distraction from this extra sensory input that's somehow making their vision work less well. Is anybody looking at that? Because I know there's like shipping lanes and and things like that. Anybody doing that in the marine world?
I would have loved to see that talk. I think so. The two reasons right whales are dying is from ship strikes and entanglement. And so alarms off of ship bows and so forth have been used to try to deter them. So this interaction of at least an auditory cue at a distance and then a visual cue up front. And what you're talking about, how sound is blocking or change, I don't know the details, but changing visual response or visual ability is a fundamental neurobiological principle called gating, where one sensory system modulates the function or abilities or responses to another. And so that's fascinating. He's seeing that at like a behavioral level. Mm-hmm. Yeah.
Oh, here's another question. Is there any species we are aware of today that we are seeing them lose their cone cells?
Armadillos, I mentioned. So yeah, we have these whales, and we've found this also in xenarthan animals, including the armadillo, but not that many. Also within non-mammalian vertebrates, so fishes. Deep sea fishes that live in the very deepest parts of the sea. We know those animals have rod-only retinas. So it was so novel for mammals.
Okay. I think we might have gotten through all the questions. Anybody else have anything they want to? Didn't miss any, did I? Anybody got any last minute questions? All right. Well, I really appreciate it. That was fascinating. And you talked about a couple of things that I used to do in my animal physiology class that I think I might have to dust off and put back in.
Do it. That's it. I'm teaching animal physiology next semester. I can't wait. It's gonna be great.
Yeah. Yeah. Awesome. Yay. Well, thank you very much.
All right, thank you. Yes, anybody feel free to contact me. And again, thank you so much for this honor. I hope you have a good evening.
All right.